nu este de a vorbi eu sau de a face, eu știu, prezentări în extenso, ci de a-l asculta pe domnul profesor Rubinsky, care, în viziunea mea și nu numai, este considerat a fi un savant. Voi da doar câteva date despre domnia sa, și anume, s-a născut la Timișoara, deci în România, a urmat școala elementară la Timișoara, a făcut licența și masteratul la Universitatea Technion în inginerie mecanică din Israel. A continuat studiile în Statele Unite ale Americii la MIT, unde în 1981 a obținut titlul de doctor inginer în inginerie mecanică, bioinginerie. După obținerea titlului de doctor, domnul profesor Rubinsky a devenit cadru universitar la Berkeley University, unde se află și astăzi. Distinsul profesor a obținut de-a lungul anului nenumărate premii. Vă spun foarte sincer, în fiecare an a obținut câte un premiu. Aș da doar câteva exemple. În 1985, din partea ASHRA, Certificate of Appreciation for Contribution to Educational Excellence. În 1994 este Fellow al American Society of Mechanical Engineers, ASME, vestită ASME. În 2013, ASME Heat Transfer Division 75 Years Anniversary Medal. În ultimul premiu, pentru că ar însemna să enumăr o listă întreagă, așa cum am spus, în fiecare an câte un premiu, și cel mai recent premiu în 2023, un premiu extrem de important, premiul Hatcher Lisner Medal al aceleiași ASME, de American Society of Mechanical Engineers, în bioinginerie. Distinsul profesor Rubinsky, dacă ar fi să raportăm toată activitatea la cifre, are un număr impresionant de citări, 31.450, și indicele Hirsch pe Google Scholar 95. Eu mă voi opri aici și dau cuvântul distinsului profesor. Îi mulțumesc foarte mult pentru onoarea pe care ne-ați făcut-o și aveți cuvântul. Mulțumesc, domnule profesor.
Mulțumesc foarte mult pentru invitație. Îmi face mare plăcere. De altfel, în ultimii ani, cred că am fost în fiecare an vreo 2-3 ori în România. O să dau prezentarea în engleză, deși, cum ați spus, eu m-am născut la Timișoara, tata e din Basarabia, mama din Bucovina și soția din București. Așa că noi vorbim românește acasă, dar engleza, româna științifică e mai slabă. Mi-am dat seama că anul trecut l-ați invitat pe profesorul Adrian Bejan. Curios, eu toată cariera mea, eu sunt după el. Am ajuns la MIT 2 ani după el, am ajuns la Berkeley 2 ani după el, dar el a continuat în energie și am continuat în biotechnology. Așa că prezentarea mea astăzi va fi în special aspecte de biotechnology. O să preluau acum screen cu permisia dumneavoastră. Cred că trebuie să mă lăsați să intru.
Sigur, Cristi, poate poți să ajuți, te rog.
Da, este în partea de jos, share screen, este activ pentru domnul profesor.
Da, share screen, sunt pe share screen, dar primesc host disabled participant screen sharing. Așa că trebuie să to allow shear screening.
Acum este în regulă?
Da,
Ok.
acum. Ok, cred că am intrat. Vedeți prezentarea?
Da.
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 Bine.
Se vede.
Da.
Din nou, mulțumesc pentru invitație și îmi face mare plăcere. Și prezentarea mea se adresează subiecte de cercetări în bioengineering. Eu voi prezenta câteva aspecte ale cercetării mele. Voi începe cu medical imaging, pe urmă voi continua cu electroporație și pe urmă cu cryopreservation. Eu am lucrat la un număr mare de tot de lucrări în inginerie, and now we'll move to English. So I worked on a large number of interesting topics in engineering. For example, I was involved in the plasma arc welding of the space station on the normal and zero gravity and energy storage in fluidized beds. And I did quite a bit of work on plasma arc welding and turbulent modeling. But today I will talk about different aspects of bioengineering. And I will start with my work on medical imaging. My research in medical imaging began in about 1980, and it had to do with cryosurgery. In cryosurgery, you insert a needle that's cooled with a cryogenic fluid in the middle of a tumor, and you freeze the tumor in the hope that the freezing will destroy the tumor. And cryosurgery was developed first in 1800s, and it's a well-known technology, but the problem was that when you treat inside the tissue, you don't see the extent of freezing. So you have no real-time control over how much you freeze. And we engineers like to have real-time control. And therefore, cryosurgery was used mostly for dermatology and outside the body. And then I started working with Dr. Gary Onek, and at that time in 1980, the technology of ultrasound was just introduced, and the ultrasound works by sending an acoustic wave, a pressure wave, into the tissue, detecting the return of the wave, and then by calculating the speed of sound, you can determine the structure and the heterogeneities inside the tissue. That's how ultrasound works. And then it occurred to us, working essentially with Dr. Onik, that we could use this technology, and this technology just appeared at that time. We can use this technology to image in real time the extent of freezing inside the body, And this has completely changed the field of cryobiology. And this is actually what we saw. This is one of the first images that we saw of freezing. You can see here, this is the margin of the liver, and this is the extent of freezing. And we could see, and it literally changed the field of cryobiology. This is actually the first patients that we treated with ultrasound monitored cryosurgery. And again, right now, everybody who does cryosurgery with medical imaging. We also received one of the first patents on that. And right now, tens of thousands of patients are treated with ultrasound monitored cryosurgery. It's inconceivable to do cryosurgery without imaging. Now, the nice thing for us engineers is that the physical properties of ice and water are so different that actually all medical imaging technologies can be used to image cryosurgery. So the next technology that I have developed is MRI monitored cryosurgery. MRI detects proton spins, and freezing traps the photons in place. And as far as MRI goes, it is detected as voids. So an MRI image obtained, this is, for instance, one device that we built in which it has an MRI coil and it's cooled by liquid nitrogen. And we've used this, for instance, to do a first study on cryosurgery in the brain. And you can see how nicely the MRI provides the extent of freezing in the brain. And we also could monitor with MRI the outcome of the procedure. And we use that for prostate cancer. I received a patent on this technology. And actually, many large MRI companies, for instance, the Sigma of General Electric, were built to take advantage of imaging of MRI. So this is another technology that we have developed for cryosurgery. This is an interesting aspect. I was working also on animals that survive freezing. This is a particular frog from northern Canada that in the winter can freeze completely and then in the spring comes back to life. So I decided to look with MRI what's happening to this frog. How come that it can return to life after being frozen? And this is what I see now. I'll explain slowly what's happening. You can look at this area first. You can see that it becomes dark. And essentially what's happening by a random nucleation, ice begins to form in the limbs of the frog. As soon as ice begins to form, look at this area. This is the liver of the frog. The liver of the frog does what everybody does when you get scared. It produces glucose, produces large properties of glucose, and this glucose acts as a cryoprotectant. And as the freezing propagates inward, more and more amounts of glucose are being produced that protect the liver, the heart, and I'm not going to show the brain, but also the brain. So by temperatures of about -5 degrees centigrade, the exterior of the frog is frozen, but the key tissues are protected by high concentration of glucose. Now, when thawing begins, the high concentration of glucose cause the interior organs to thaw first. And then the blood starts functioning, and by the time the limbs freeze, the entire blood circulation is renewed. So that's an interesting application of MRI. The The other application was with David Van Aaron and David Oten. We realized that pretty much every physical process, essentially energy delivery, can be used for imaging. And we went and we used light to determine changes in the frozen media, and we injected various frequencies of light and monitored the path. And with that, we were also able to monitor freezing. And again, we got a patent on this technology, and a company was developed on the basis of this technology. The other interesting aspect of imaging is electrical impedance tomography. In electrical impedance tomography, you inject milliamp currents inside the body and record voltages from various electrodes. And together you develop, and this is pretty much how it looks, you develop an algorithm that essentially by solving the Poisson equation, you can map the electrical impedance inside the body. And this is a very interesting technology. And again, we have shown that we can monitor freezing with electrical impedance tomography, again, because ice essentially does not conduce electricity while the surrounding tissue does. I started working quite a bit in electrical impedance tomography. And the interesting thing is that the amount of raw data is very small, just a vector of currents and voltages. So it occurred to us that we can actually use telecommunication to transmit the data to a data processing facility and then reconstruct an image. And because data processing is very heavy, but the amount of data is very small. So, for instance, this is a simulation of a breast cancer tumor, and this is the image of the breast cancer tumor that we obtained by sending the data with a cell phone and reconstruction back. And again, we received a patent for this concept, and I believe that this is the first patent on medical imaging through the internet. And so what we found is that in many imaging technology, the data acquired is small, and the image reconstruction takes a substantial effort. And this is actually a paper that was featured in Nature at the time. But we have shown that with a simple ultrasound acquisition device, a two-dimensional one, we can reconstruct a three-dimensional image. Right now, there are quite a few companies that use these handheld ultrasound devices, homemade, but we are one of the first, actually. We have the patent on this. My current research on imaging essentially focuses on the interaction between electrical currents and imaging, the electrical impedance tomography technology. And I'm working with Professor Cesar Gonzalez in Mexico. And what we realized that internal bleeding is not being recognized or head injuries. And we developed breast cancer is also a major problem. So we realized that it is sufficient to have inexpensive diagnostic at a small village to use for triage, and then the patient can be brought for MRI or CT or so on. So we've developed a simple device in which essentially there are two coils, and an electromagnetic signal is transmitted between these two coils. And we're looking at phase shift to develop diagnostics. And this is, for instance, this is one of our devices, but this is a very interesting experiment that we have done in Mexico. We put the coil on patients that are healthy, that had edema in the brain or a hematoma in the brain. And we plotted the results as a function of age. And you can see here, and this is an integration of particular frequencies. And you can see here how the measurement changes with age, and the people with hematoma or edema fall on this curve. The interesting result is that after the age of 70, your brain has the signature of somebody with edema or hematoma. So bad news for us. Anyway, we've developed a classifier, and this classifier can tell you if you have edema or if you have hematoma, and it actually is being used now clinically. It has CE mark approval and FDA approval for detecting the brain. Now let me move to electroporation technologies. Electroporation, like the name implies, is it has been observed that when a particular electric field is applied across a cell of magnitude of about 2000 2000 volts per centimeter, and very brief, in microseconds, the cell membrane becomes permeabilized and pore forms. This is the source of the name electroporation. And now let's go back to another topic that I worked is, I went to Antarctica and I've studied Antarctic fish. And the Antarctic fish, we discovered, have a property that they protect the cell membrane. This is a oocyte, an egg of pigs, and from the effect of cold. So this is a pig egg in cold, and this is a pig egg protected with antifreeze proteins. And this again resulted in a patent and in 2 applications. One of the most successful ones is in cosmetics. So if you buy expensive cosmetics, you may have AF protein. The other one was essentially in genetic engineering. We realized that when you apply that, you can use the so-called gene that produces antifreeze proteins to bind to a growth hormone, and you can use that to raise a salmon that grows the entire year long. So that was the first animal, transgenic animal, and this particular salmon has now FDA approval and is being sold. And the advantage is that it can grow the entire year long, whereas normal fish cannot grow the entire year long because in the winter they have no food. One of the big complaints of the FDA was what's happening if the salmon escapes in the wild. So I decided, and there were no models for that. So I decided to use actually the principles of energy, conservation of energy, to study genetic evolution. And you'll see how analog there are one to each other. So if G is a vector of genes, like a chromosome, you have a tensor that manipulates these genes and produces so-called a phenotypic type. So essentially, we are a gene on which a tensor operates, and that's how we look. So this probably should remind you of something, but if not, I'll show you the equivalence to energy equation. And the second component is this phenotype. If you operate on it with a particular, again, another matrix, you can generate a fitness. So somebody that runs faster is more fit. So you can essentially correlate mathematically between the phenotype and essentially the fitness. Well, this looks very much like the relation between heat flux and temperature. Heat flux is essentially an operator, thermal conductivity on temperature. The fitness of the solution to the Laplace equation is analog to conservation of energy. So essentially, you can determine the fitness of a solution to the Laplace equation by looking at how well the solution satisfies the conservation of energy equation. So how do we solve the Laplace equation? There are various means, but I looked at the finite difference scheme in which you have temperatures at different nodes, and these temperatures essentially can be described as a vector. This vector looks very much like a gene, with each loci of the gene having a different value. And I worked with parallel computing of up to 40,000 elements. So what we did, we essentially manipulated in similarity to gene evolution. We began by using random generation to generate a solution to the energy equation at each node in the form of a random generative vector with temperatures, essentially. Then we evaluated the fitness of the solution, essentially how well it satisfies the energy conservation, and gave it a value from 0 to 100. Then we went and applied reproduction to this. Essentially, parent 1 with parent 2 exchanged parts of their gene at random locations. And we gave the vector with a higher fitness more probabilities to procreate. So, and also from time to time, the computer introduced a random mutation, and then we let the system grow. And these are the results. We studied evolution, and these are the number of generations. Something interesting, these are the highest fitness, these are the lowest fitness, these are the average fitness. And what you can see that within 20 to 40 generations, any organism reaches steady state for particular outer conditions. It will never reach the maximum. There will always be somebody that's maximum and somebody that's minimum. We apply that to what's happening if a fish escapes in the wild. And we correlated that to the viability of the transgenic fish relatively to the wild type. And we found that as a function of, and this is the plot of time to extinction. And what we found is actually that for certain conditions, the wild type will not get extinct. And that was very important for the FDA because they were concerned that if a fish escapes in the wild, the normal type will get extinct. But we've shown that because the transgenic fish will die in the winter because it has no food, then after a few generations, it will disappear. And this is how it essentially looks. At first, there is a large frequency of transgenes, but then very soon it disappears, and then the normal population grows back. And this was 10 individuals in a population of 1,000, but the ratio is much smaller. We also played around and looked at the effect of eugenics. What's happening if you kill the minimum fitness individuals? Well, if you do that, and if you remove all this all the simpletons, you will never reach a maximum. So you cannot use eugenics. Now let's go to electroporation. And as I mentioned, in electroporation, you apply a pulse across a cell, and then the cell membrane opens. And then you introduce a gene. There are parameters in which called reversible electroporation, in which the cell membrane closes after the electroporation pulse is stopped. And there are conditions in which the cell membrane does not close. In genetic engineering, and this is going back to the transgenic fish, we've developed technologies to genetic engineer the cells using electroporation. The problem was that there was no real-time control over the process of electroporation, and what I've developed is a device in which there is a MEMS device with a micro hole, and this is the cell trapped between top and bottom chambers. And my idea was that when you apply an electric pulse, and if the cell membrane opens, then there will be a current going through the cell, and then you can detect in real time if the cell has become electroporated or not. And we've used that for controlling electroporation. And actually now there are many companies that are using this technology. This is the first genetically engineered cell with this technology. So this technology, essentially single cell electroporation, is one of the first MEMS devices with essentially with living cells, and it was featured in Nature, and as you can see here, and essentially it's now one of the standard methods for electroporation, and we got 1 billion patents for this. Not done anything with it commercially because other companies really developed the technology, but Berkeley still holds the patent. Then, if you remember, I mentioned that there are parameters that destroy the cell membrane and the cell dies. So over the years, I have worked on many technologies for tissue ablation for treatment of cancer, cryosurgery, but also microwave treatment and ultrasound, radio frequency. All these technologies are being used to destroy cancer. The problem with these technologies is that when you apply heat or cold, you essentially destroy all the molecules in the tissue, including blood vessels. And it occurred to me, working with my PhD student, that if you were to apply the so-called irreversible electroporation on the tissue, then the only thing that will happen is you'll affect only the cell membrane, and all the molecules will remain intact. So it occurred to us that if we can design a device that applies irreversible electroporation pulses, and the device is simply 2 electrodes introduced into the tumor, and then you will actually spare the blood vessels, the nerves, and the only thing that will die is the cell membrane that essentially is irreversibly permeabilized with pores. So, of course, because we are mechanical engineers, we developed a mathematical model to calculate if it's possible to apply energy at the level of irreversible electroporation, but without causing thermal damage. And actually, the first work in the field of irreversible electroporation was mathematical. We've demonstrated that there are certain particular electric fields that can cause irreversible electroporation, but do not cause thermal damage. And then we began working. I worked also with Marcus and Gary Onek. And we started the work in 2005, and in 2008, we got already the FDA approval. We sold the company to AngioDynamics, and now our device is known as NanoKnife. And at this stage in 2008, everyone else entered the field, and we've demonstrated lots of valuable applications. This is, for instance, in the liver. This is a pig liver. we applied irreversible electroporation, and you can see what's happening. It looks dead. And then I think it's 3 days later, you can see the liver begins to regenerate. The dead cells are removed by the immune system. The liver regenerates. In 2 weeks, it's completely regenerated. And the blood vessels inside this tissue remain intact. That's of great value in treatment of cancer, because until now, it was impossible to ablate, to destroy cancer near large blood vessels. You see, that's why you cannot, it was impossible to treat pancreatic cancer. Also, from our past experience, if you use thermal ablation, what you get here is scar tissue after 2 weeks. It's not a normal liver. Here, the normal liver became completely regenerated. Again, for example, this is a liver and this is a tumor around a large blood vessel. It was impossible to treat this tumor before. With non-thermal irreversible electroporation, you can just go and treat. You can see here ablation around the blood vessel, around bile ducts. And this has made a huge change in the treatment of cancer. There are now thousands of researchers working in this field that we have invented, and we have the first patent on it. Lots of applications. One of the key applications is for treatment of atrial fibrillation. Many of the biggest companies in the world have entered now into doing treatment of atrial fibrillation with irreversible electroporation, but this is the first paper that was ever written on this concept. Medtronic and pretty much St. Jude's, Boston Scientific, they all entered into this field. Now, this is actually the first clinical paper. We published the first use of irreversible electroporation for treatment of cancer, prostate cancer, and we've shown that the nerves remain intact after treatment of cancer with non-thermal irreversible electroporation. And essentially, this is the first series of 16 patients with excellent results. And again, there are hundreds or thousands of patients now treated for prostate cancer with irreversible electroporation. Now, perhaps the most impressive result is that it's finally a method for treatment of pancreatic cancer. Again, it has received FDA approval. It is used all over the world now for treatment of pancreatic cancer. You could not treat pancreatic cancer neither by resection nor thermal, because it's perfused with blood vessels. With this technology, you can treat pancreatic cancer. So it's finally a surgical technique for treatment of pancreatic cancer. What am I doing now in this field? Actually, I noticed that, as you have seen in the previous slides, that the liver can regenerate after treatment with irreversible electroporation. And why can it regenerate? Because the so-called extracellular matrix remains intact. So irreversible electroporation destroys only the cell membrane. The immune system removes the cell membrane, but everything else that is needed for a cell to attach and regenerate keeps working. So what we've done with Yanfang Zhang, we just published, I think, this year. We treated a liver with irreversible electroporation, and then we inserted pancreatic islets in the region of the liver that was treated with irreversible electroporation. And we found that the pancreatic islets became incorporated in the liver. And this is a potential treatment for diabetes type 1, because the insulin has to go to the liver anyway from the pancreas. So with this technology, the insulin is produced in the liver, and we have shown that the pancreatic islets that we implanted in the liver produce both insulin and glucagon, which is essentially what is needed for treatment of diabetes type 1. And we may have a treatment for diabetes type 1, a very simple treatment. It's essentially, it doesn't require surgery. You just introduce 2 needles for electroporation, and 3 days later, you inject the pancreatic islets, and essentially the patient begins to produce insulin and glucagon. And of course, we got the patent for this. Recently, again, just several weeks ago, we found something extremely interesting. We were able to use irreversible electroporation to ablate a liver that was fibrotic. Essentially, the problem of cirrhotic liver, essentially. In cirrhotic liver, there is no other treatment except liver transplant. What we have done is we have treated the liver, the cirrhotic liver, with irreversible electroporation, and after the immune system removed the damaged cells, the cirrhotic cells, we implanted normal hepatocytes, and these grew. So it's possible that this technology could be also a solution for fibrotic and cirrhotic livers. So I have about 20 more minutes, and then I'll discuss the current research in cryopreservation. So perhaps the most exciting work that was done primarily in collaboration with Romania and with Professor Alexander Shevan and Gabriel Nastasis is the concept of isochoric cryopreservation. Now, the principles of isochoric cryopreservation are the following. Most research in life sciences is done in isobaric, atmospheric, constant pressure systems because it's natural. And it occurred to me to see what is going to happen if you study biological processes in constant volume systems. And of course, because most of my focus is on cryogenics and low temperatures, I wanted to study what's happening if you do isochoric freezing of biological materials. And what emerged is that if you freeze an isobaric atmospheric pressure, this is the phase diagram of ice and water, as soon as you pass the liquidus line that separates between water and ice, the entire volume will freeze. And the freezing of the volume is actually the main mechanism that destroys biological tissue, that makes it impossible at this stage to preserve whole biological organs. However, in a constant volume system, when you have nucleation, this is a simplistic explanation. We have a more fundamental on the basis of Helmholtz equilibrium. If ice forms, ice has a lower density than water. And when ice forms, the ice expands, and then there is an increase in pressure in the system. And we've shown that in an isochoric system, the freezing process goes along the liquidus line. So as you freeze, you don't enter into the one-phase domain. You always stay in the two-phase domain. And the two-phase domain is ice and liquid. And at the triple point, minus 21 degrees, about 22 degrees centigrade, the volume of the liquid phase is about 50%. So up to the triple point, a large percentage of the liquid remains unfrozen. So if you introduce biological matter in the unfrozen part of the liquid, then that biological matter will not freeze up to minus 22 degrees centigrade. And again, to repeat, the main problem with cryopreservation of biological organs is the formation of ice inside the organ. That's what destroys the organs. Single cells you can protect from ice damage by adding various cryoprotectants, glycerol, ethylene glycol, and so on. But you cannot do that in a whole organ. So until now, there was never a study that demonstrated that whole organs can be preserved at temperatures below freezing because ice forms. In isochoric freezing, ice is not going to form. So it removes an important aspect of damage. So this is actually the curve that shows the percentage of ice as a function of temperature. Again, obtained patents. I make it a rule, although I'm not using most of the patents, but I'm making the rule that whenever I have something new, I apply for a patent through the university. So this is I would say, probably the most exciting finding. We were successful to preserve biological organs at sub-freezing temperatures, a whole liver, a whole pig liver, and the first ever isochoric supercooling preservation of an organ was done in Romania with the collaboration of, and primarily with Professor Anastasis and Professor Sherban. We have found, in addition to the effect of isochoric freezing on the process of freezing itself, we have also found that in the isochoric conditions, you stabilize the supercooled thermodynamic state. Essentially, the same effect of expansion of the ice nucleus when freezing begins has also an effect on stabilizing the thermodynamic supercooling state. What does it mean? A thermodynamic supercooling state is metastable. You know that you can reduce the temperature of water, of pure water, up to minus 40 degrees centigrade without it freezing. Of course, it has to be pure and small volumes. But ideally, if you would be able to supercool the system without any ice forming, then there is a substantial advantage in preservation. Why? Because when you do isochoric freezing, the pressure also increases. And below certain pressures, like 100 megapascal or so, the pressure begins to negatively affect the organ itself. If you just supercool, then there is no effect, no increase in pressure, and you can go to lower temperatures without an increase in pressure and without formation of ice. But supercooling is an unstable state. What we have discovered, and this is a recent paper, again, led by the group of Gabriel Nastase and Alexander Sherban, we have shown that biological solutions are stabilized in the supercooled state. Again, the mechanism is that when an ice nucleus forms, the pressure acts against the growth of the ice crystal. so-called Le Chatelier effect, and therefore it stabilizes the metastable state. I believe that this is an important finding, and Physics of Fluid is probably the most well-recognized journal in the physics of fluids. So we have demonstrated first theoretically that supercooling is stabilized. Another mechanism is mechanism of vitrification. Vitrification is the formation of a glass. The glass, you can form a glass if you increase the viscosity of water to high levels, and this is usually done by lowering the temperature to liquid nitrogen temperature very fast. But it turns out that isochoric systems facilitate vitrification. I will not go into this. This is a patent that was issued with Gabriel Nastas as a co-inventor in that patent. Let us just go to isochoric supercooling. So we decided, again, with a group of Sherban and Nastas, Irinel Popescu, Florin Botia, to try to apply isochoric supercooling for a whole liver preservation. This is the system that was designed by the group of Professor Sherban for isochoric supercooling that essentially contains an isochoric chamber in which you can monitor pressure and temperature, that can be immersed in a cooled bath, and the content of this, we have shown, can reach sub-zero temperatures without the organ freezing. This is a very large volume. In the absence of isochoric conditions, this volume would freeze at sub-freezing temperatures. In isochoric conditions, this volume does not freeze. And this is essentially the result, again, with done by the group of Irinel Popescu, Professor Irinel Popescu. And we have shown that we can preserve a pig liver at sub-freezing temperatures for 48 hours without causing damage to the liver in a supercooled state. And obviously, there is huge interest in this technology. We received $25 million from NSF to develop this technology for organs, received $2 million from NASA to develop technologies for bringing biological materials in space and back. But in the past, the maximum type you could preserve a liver for transplantation was about 8 hours. We managed to preserve for 48 hours, and I'm sure it can be done, preserved much longer. But I think that this invention of the technology will be credited in the future to the work that was done in Romania with our group. And it's impressive. I can mention that we already have citations from reviews on this technology. So again, these are the technologies that we work on. Recently, it's not completed, but we managed to preserve a liver and transplant. I just cannot remove the sound, but we managed to preserve a liver, a heart for 21 hours and transplant that heart at the Shiva Hospital in Israel. And again, normally hearts can be preserved maximum 6 hours. And we're continuing with this research. So now this is the last topic, 10 minutes. The last topic is temperature-controlled cryoprinting. Now, you know that everybody's interested now in printing artificial organ. And Nature and many other journals present results for what they call a printed organ. The problem is that the organs that are currently printed are pretty much 1 or 2 centimeters in dimension. The reason is the following. Printing is extremely slow. So by the time you finish printing such a heart will die, the cells will die, and also the tissue is soft, so you cannot build a large structure, it will collapse in itself. So it occurred to me that how about if we print the organ while it is immersed in a cryo liquid? And when you do that, each individual voxel that is deposited will freeze. And the freezing, we can design freezing in such a way that you freeze the cell with optimal cooling rates. Single cells can be cryopreserved. Large organs cannot be cryopreserved. So the freezing has 2 effects. One, it provides rigidity to the organ that is printed. Second, it freezes every cell with optimal freezing conditions so that they survive freezing. And then you can print a large organ and it will survive. And this is an example, okay? This is what's happening if you print something without freezing, and this is the same structure printed with freezing. The design is the following. You have a cryogenic bath, you print a layer, you can control the freezing with a controlled freezing rate for optimal cryopreservation, and the print plate moves down layer by layer. We can also control the microstructure, which in the past was impossible. And we have also developed recently a mass manufacturing unit in which we have the 3D printer, we do cross-linking, directional freezing, and then with a robotic arm, we assemble one layer on top of the other. These are some demonstrations. So this is how we freeze a layer of tissue. This is how we cryoprint. So essentially each droplet freezes as it is being deposited. And this is the mass manufacturing technology in which the robotic arm takes each layer, deposits the layer on top of the previous layer, it freezes, and then it comes and picks up another layer. And we have also developed various non-linear printing technologies because organs are not cubes. So these are some recent results with temperature-controlled cryoprinting. We managed to cryoprint food for patients with dysphagia. Dysphagia patients are those that cannot swallow the food. So, but on the other hand, and that's what they're given, a mush. This is very detrimental to their well-being because it's unpleasant, they cannot eat in company. So we decided to use cryoprinting to provide texture to this kind of food so that they eat it as if they were eating a normal steak, but when it goes into the mouth, it becomes a mush. And this was, again, recently published. The other result that was recently published, we managed now to cryoprint structures of up to 15 layers, and the cells survive the freezing. So this is cell viability. This is the whole process that we're doing. But right now we can do what nobody else was able to do in the past, namely print an unlimited volume of tissue in which essentially the cells survive. So we hope to go and print now an artificial liver or various other cell structures. And now, again, we have the patent for this. And in summary, this is my most important accomplishment in life. My grandchildren, these are the babies, these are the girls, and these are the boys. And thank you very much.
Mulțumim!
Mulțumesc frumos că mi-ați dat posibilitatea să prezint lucrarea mea.
Mulțumim! Noi vă mulțumim foarte mult! L-am văzut pe Gabi, este logat Gabi Năstase. A fost o pledoarie cum ingineria mecanică, criogenia împreună cu medicina pot crea lucruri extraordinare. Puse împreună cu oameni extraordinari pot face progres. Gabi, nu știu dacă domnul Șerban, cred că este pe telefon cumva, nu-l văd cu numele, dar Gabi Năstase este conectat și ne mândrim că avem colegi care au avut șansa să lucreze cu domnul profesor. Întrebări dacă sunt.
Se poate să iau eu cuvântul un pic?
Da, Gabi, sigur.
Da, Gabi.
Bună seara! Da, este...
Bună
seara la toată lumea! Sunt bucuros și încântat să particip la această întâlnire și la această prezentare a profesorului Rubinsky. Eu am fost acolo la dumnealui în laborator și am învățat foarte multe lucruri din experiența pe care am avut-o în Statele Unite și bineînțeles că tot ce a prezentat dumnealui este de un nivel ridicat de calitate științifică și ați văzut și dumneavoastră câte patente, câte brevete are dumnealui în spate și pe câte zone de tehnologie și cred că este de apreciat toată această muncă și sigur că are toate meritele în spate și doresc să-l felicit încă o dată, cum o fac de fiecare dată și sunt întotdeauna impresionat de munca dumnealui.
Mulțumesc, Gabi.
Frumos, cu mare drag.
Este mare plăcere să lucrez cu tine și cu profesorul Șerban și
nouă la fel. Și nouă de
sper să
asemenea.
continuăm în viitor.
Categoric.
Să dea Dumnezeu și vin în România din nou.
Bine, vă așteptăm cu drag. Seară bună tuturor, cele bune, la revedere.
Mulțumesc, să trăiești. După cum ați remarcat, din acest colectiv face parte și domnul academician Irinel Popescu și ne mulțumim că avem colegi care sunt într-o astfel de echipaj. Mai vreau să adaug un lucru că din 19 octombrie 2015, domnul profesor este doctor honoris causa al Universității Dunărea de Jos din Galați și mă bucur că ne-a onorat cu acceptând să facă parte din comunitatea academică de la Dunărea de Jos. Dorea cineva să întrebe ceva? Sigur. Sau am auzit ceva de genul?
Păi, aveți e-mail-ul meu, puteți în telefon să-mi scrie.
Sigur.
Și sper că vin din nou în curând în România, îmi face mare plăcere.
Vă așteptăm și vă mulțumim din suflet, dorim să ne revedem cu bine și aș vrea să vă aplaudăm în final. Vă rog să nu ieșiți ceilalți invitați, pentru că vreau să fac un anunț pentru ziua de 1 noiembrie, ora nu va fi 3 p.m., va fi 2 p.m. la Victor Oancea. Să-l aplaudăm pe distinsul profesor Boris Rubinsky și să-i urăm sănătate și să-și împlinească toate visurile, nu numai în cercetare. Mulțumim, domnule profesor!
Mulțumesc frumos!
Mulțumim
La revedere!
Mulțumim!
Sănătate multă!
Mulțumesc!
Mult succes!
Mulțumim!
La revedere!
Sărut mâna!
La revedere!
Cristi, mă auzi? Da. Vă mulțumesc foarte mult. Deci, cei care
La
mai
revedere!
sărut una. Pentru data de 1 la întâlnirea cu Victor Oancea de la Simulia din Boston, am decalat ora, având în vedere și schimbarea orei de iarnă la noi în România. Deci nu va fi ora 15, va fi ora 14. O să modific și pe site. Vă mulțumesc din nou pentru participare. O seară bună și spor în tot ce faceți. Mulțumesc. La revedere.
La revedere!
La revedere,
Mulțumim.
noi mulțumim.
Mulțumesc.
La revedere!
Mersi. Cele bune. Mulțumesc colegilor mei pentru participare. Mulțumesc mult.